On an average, we inhale around 10,000 microbes per day, but these microbes are not able to colonize our respiratory system because of presence of specific nasal hair, cilia, mucosa, and specific macrophages. So, whenever air enters through your nose, it is filtered by means of these nasal hair, which removes large dust particles laden by the microbes. And those microbes, which are or dust particles, which are able to bypass these nasal hair, then enters through the pharynx, which and the trachea, which is lined by specific hair-like cells called cilia, and this cilia is covered by a layer of mucus. This mucus layer traps the pathogen or the dust particles or the pollens, and this cilia then, with the help of its mucociliary movement, it moves the dust particles or the foreign material towards the pharynx, which is then removed by means of coughing or sneezing out of the host's body. So this type of movement which removes the pathogen or the invader out of the host from the pharynx towards the mouth is called mucociliary escalator. So if the pathogen is able to pass through the trachea and enters the lungs, there are specialized type of cilia present there with them which removes the microbes and debris from interior of the lungs, and there are specific type of macrophages called alveolar macrophages which eliminate those microbes and present those antigens to the adaptive immune system. Let us see the immunology of lungs in detail with the help of this video.
The lungs are where the body draws in vital oxygen and exchanges it for carbon dioxide. But every breath we take is accompanied by potentially harmful microbes, as well as particulates which can damage delicate lung tissue. To counter these threats, the lungs are enriched in immune cells that repulse invaders and repair injury. But this potent arsenal comes at a heavy cost. Overreactions or inappropriate responses result in diseases such as asthma. The lungs are spongy organs composed of increasingly finer tubules. First comes the trachea and the bronchi. These are then divided into the bronchioles, which are wrapped in a muscular sheath. At the ends of the bronchioles are microscopic air-filled sacs called alveoli. In the case of asthma, most of the harmful immunological events occur within the bronchi and bronchioles. The bronchial wall is made up of an epithelium covered in tiny hair-like structures called cilia, an interstitium, and muscle cells. Goblet cells scattered along the epithelium discharge a thin layer of mucus into the bronchiole lumen. The mucus is continually swept back up through the lungs by the action of the cilia. This system is known as the mucus escalator and is important for removing inhaled particles trapped within the mucus. Embedded throughout the bronchiole wall are immune cells such as dendritic cells, which sample antigens from the lumen, mast cells, which are packed with granules, tiny packages rich in histamine, and innate lymphoid cells, or ILCs.
The gastrointestinal tract is the primary site of interaction between the host immune system and the food-derived antigens and toxins. So this mucosal immunity begins at the epithelial cell of the gastrointestinal tract, which prevents entry of most of the microbial pathogens. Moreover, the acidic pH of the gastric juices, the presence of pancreatic enzymes, bile salts, and gastrointestinal associated lymphoid tissue keeps the number of population at check and prevents the colonization of most of the pathogenic bacteria and other pathogens. Moreover, the unidirectional peristaltic movement of the intestine also helps in preventing entry of microbes from the dense distal gut content to the small intestine. There are specific types of flora present in the gastrointestinal tract called the normal flora, which also prevents the pathogenic bacteria colonization at specific sites. There are special types of cells called Pennant cells which are present in the small intestine which secrete specific antimicrobial peptides such as lysozymes and tryptophan which cause lysis of the microbes by affecting their different membranes and cell wall. Most of the microbes are not able to colonize the genitourinary tract due to a low pH or hypotonic condition in the kidney and presence of specific type of microbes in the genital tract like lactobacillus acidophilus which keeps the pH to a lower level like around 3 to 5 at which most of the bacteria cannot colonize or survive. The eye has specialized mucus-secreting epithelium membrane which secretes specific mucus which contains enzymes like lysozymes which degrades the bacterial cell wall, lactoferrins which prevents the free iron and make it unable to be used by the microbes and leading to their death, and secretory antibodies like IgA. Together, all of these help in prevention of colonization of most of the microbes. Chemical barriers forms the first line of defense and they prevent the colonization and invasion of most of the pathogenic bacteria. Let's look at different types of chemical barriers which are present in the human host. First is low pH, which prevents the colonization of the pathogen in the vagina, in the urinary tract, and in the stomach. The second type of chemical barrier are chemicals called defensins, which are basically short, like 29 to 35 amino acids, antimicrobial peptides, which can form pore into the pathogens or bacterial membrane, and they are basically of two types, either alpha defensin or beta defensin in humans. So alpha defensins are generally secreted by the neutrophils and the macrophages and by the Pennant cells in the intestinal tract. However, the beta defensins are secreted by the epithelial cell of respiratory tract and urinogenital tract and most of the leukocytes. So these defenses have a broad spectrum of action, so they can show antimicrobial activity against gram-positive and gram-negative bacteria, certain types of fungi and viruses and protozoans. So third type of chemical which provides defense against pathogens are lysozymes, which causes degradation of the cell wall component of bacteria, that is peptidoglycan. This chemical is present in the saliva and the tubes. And the fourth type of chemical is bacteriocins, which are basically small peptides or proteins which are produced by one bacteria against another type of bacteria. So colicins are the bacteriocins produced by E. coli and cephalococcins are produced by Staphylococcus. So these are cationic in nature generally and they cause change in permeability of the cell membrane of the pathogens and lead to their death. Another type of chemical barriers are beta lysins. These are produced by the platelets during coagulation and these are basically amino acids and they causes lysis of the gram-positive bacteria by acting as a cation detergent. Normal flora is also part of the first line of defense of innate immune response. So these normal flora consist of microbes which are present in specific sites of our body which generally don't cause any disease in the host, as we have discussed in our clinical microbiology lectures. So these normal flora provides competition to the pathogenic bacteria and prevents their colonization by eliminating the essential nutrients and utilizing them. And secondly, they can also prevent their colonization by releasing specific inhibitory substance. Like we in the last slide we have seen, they produce some specific chemicals called bacteriocins which kill the other bacteria. So by the help of both these features, they are able to reduce the number of pathogens and eliminate them. Now let's have a look at how the normal flora provides immunity. So students, it's time for our second conceptual point. So in the first question, you have to enlist all the type of barriers which Vibrio cholera has to bypass in order to colonize the intestine in humans. In the second question, you have to find out whether there is a difference in the normal flora or microbiome of babies born by normal birth and C-section. Your answer should also indicate why and how, if it varies or not. Try to answer these questions and we will discuss them in our interactive session.